What if the future of dentistry is not a plastic implant, not a porcelain crown, and not even a root canal, but a living tooth grown from your own body? Not a fake replacement, not a cold metal post. A real tooth with living tissue, blood supply, and maybe even the kind of natural connection that makes it feel like it truly belongs to you. That sounds like science fiction, but the newest conversation in regenerative dentistry is starting to move in a surprising direction, away from stem cells alone, and toward the hidden power of glial cells. And that raises a big question. If teeth can be replaced today with implants and dentures, why are scientists still chasing something more? Because modern solutions are useful, but they are not the same as a living tooth. Dental implants are metal posts placed into bone, and dentures are removable. They can restore appearance and function, but they do not bring back the full living tooth system, including the natural pulp, nerves, and ongoing repair capacity that healthy teeth have. That difference is exactly why regenerative dentistry keeps pushing forward. For years, stem cells were seen as a superstar of tooth regeneration. And to be fair, they still matter a lot. Scientists have shown that dental pulp stem cells can help regenerate pulp-like tissue, support nerve repair, and even promote tissue healing in experimental settings. But there has always been a problem. Building a tooth is not just about making cells. It is about making the right cells in the right place at the right time, with the right blood supply, nerve supply, and structure. In other words, a tooth is not a pile of bricks. It is more like a city with roads, power lines, plumbing, and a blueprint that all have to appear together. So what exactly are glial cells? In simple terms, glial cells are the nervous system's support crew. They are the helpers, protectors, and organizers around nerve cells. In the peripheral nervous system, the best-known glial cells are Schwann cells. You can think of them like a repair team and insulation at the same time. When nerves are injured, Schwann cells can switch into a repair mode, help guide nerve regrowth, and release signals that support healing. That is a big deal, because teeth are not isolated blocks of calcium. They are closely connected to nerves, and that connection matters more than most people realize. The dental pulp inside a tooth is richly innervated and vascularized. That means it has a strong network of nerves and blood vessels. Those nerves are part of why tooth pain exists, but they also help maintain the health of the tooth tissue itself. In simple language, the nerve supply is not just there to warn you when something hurts. It is part of the living system that keeps the pulp active and responsive. Now, here is where things get really interesting. Recent research suggests that the nerve environment inside and around teeth may be a key missing piece in regeneration. Scientists are studying how Schwann cells and Schwann-like cells can support sensory nerve regrowth in dental pulp repair. Some work has even explored turning dental pulp stem cells into Schwann-like cells, because sensory nerve regeneration in the pulp is one of the hardest parts of true healing. That means glial cells are not just background actors. They may be part of the repair script itself. So why is this such a big breakthrough idea? Because the old stem cell approach often focused on building tissue first and hoping the rest would follow. But a tooth does not grow well in isolation. It needs nerve signals, blood vessel growth, and a supportive microenvironment. Glial cells may help coordinate that environment. They can support axon growth, help repair damaged nerves, and may indirectly influence how dental pulp cells behave. If stem cells are the raw construction material, glial cells may be part of the foreman team that tells the construction site what to do. That is why researchers are becoming more interested in nerve-guided regeneration instead of tissue-only regeneration. Some studies already show that dental pulp cells can promote peripheral nerve repair, and other research suggests that Schwann cells are essential for successful nerve regeneration. There are also newer experimental studies using dental pulp stem cells, nerve conduits, and Schwann cell-related strategies to improve regeneration in animal models. None of this means we can walk into a clinic tomorrow and order a brand new living molar, but it does mean the biology is becoming more believable. Still, let's stay realistic. There are real hurdles. Tooth regeneration is not just about getting cells to survive. Scientists still have to solve tooth shape, root formation, enamel development, blood vessel integration, nerve integration, immune response, and long-term stability. Reviews of regenerative endodontics continue to say that true pulp regeneration is still far from fully achieved in routine care, and that clinical translation remains challenging. So yes, the science is exciting, but no, this is not a miracle that is ready for every patient right now. Even so, the progress is real. Researchers have already reported bioengineered tooth buds in animal models, including work in mini pigs using Other studies have explored regenerative teeth formed from in vitro mesenchymal cells in experimental systems. These are early steps, not final products, but they show something important. The idea of a biologically grown tooth is no longer fantasy. It is an engineering problem that scientists are actively attacking from multiple angles. So what might dentistry look like in 10 or 20 years? The most realistic future is probably not full replacement teeth for everyone overnight. It is more likely that we first see better pulp regeneration, stronger root repair, smarter biologic fillings, and partial tooth restoration that keeps teeth alive longer. After that, maybe we move toward lab-grown tooth buds for selected cases. And if glial cells really prove to be the missing switch, they could help turn a damaged tooth site from a dead repair zone into a living regenerative environment. That would be a huge shift. Imagine this. One day you lose a tooth, and instead of an implant screw, a lab-grown living tooth is guided into place using your own cells, your own nerve-supporting biology, and the body's natural repair signals. It would not just look like a tooth. It could behave more like a tooth. That is the dream driving this field forward. Not hype. Not magic, just very clever biology trying to solve one of dentistry's oldest problems in a completely new way. So here is the real takeaway. Glial cells may not be the whole answer, but they could be a crucial part of the answer. They may help scientists unlock nerve repair, pulp regeneration, and maybe one day true living tooth replacement. And that makes this one of the most exciting ideas in modern dentistry. Would you trust a lab-grown living tooth over an implant? If you enjoyed this video, hit like, subscribe, and comment your answer below.